Welcome students, today 8th Science, Unit 8, Unit 16, Microorganisms, today topic, microorganisms. Microorganisms means very small organisms. Microorganisms are very small in size. They cannot be seen our naked eye, only with the help of microscope we can see the microorganisms. So this microorganism is called microbes. Okay, the microbes, study about microbes, study about microorganism is called microbiology. Microorganisms are found everywhere. Microorganisms found everywhere. They are found in air, water, soil, and even inside our bodies. In water, ponds, lakes, rivers, and oceans. They remain inactive under unfavorable conditions and become active during favorable conditions. In unfavorable condition, the microorganisms are inactive. They cannot multiply. They cannot get growth months. They cannot do any active function. It's inactive. But the conditions are favorable. It's become active. Can multiply and get growth months. So microorganisms can be studied under 5 categories. They are virus, bacteria, fungi, algae, and protozoa. Let's study about virus. Let's study about virus. Virus is a tiny particles, very smallest particles made up of genetic material and protein. Genetic material may be a DNA or RNA. They are intermediates between living and non-living things. So the microorganism exposed to the virus, it's showing two characters, living characters and non-living characters. When inside the host cell, it's showing that living character. When it is outside of the host cell, in the environment, when it is present freely in the environment, it's showing the character non-living characters. So virus means poison, the Latin word. Viruses are intercellular obligatory parasites. Parasitic organisms depend on other living organism for its food and shelter. The study of virus is called virology. The study of microbes is called microbiology. The study of virus is called virology. Viruses are 10,000 times smaller than bacteria. Viruses have different shapes, rod shape, spherical shape, or of other shapes. The virus, it contains code, the DNA or RNA. The virus may be a DNA or RNA. So you can see here the DNA virus. This is RNA virus. So virus contains DNA or RNA. This DNA or RNA surrounding the coat is a protein coat. In some virus, the protein coat is covered by an envelope made up of protein, lipids, and carbohydrates. So these envelopes have spikes. The spikes. So this is a spike. So this spike is helped to attach the host cell. This spike is helped to attach the host cells. So these envelopes have spikes that help the virus particles attach to the host cells. Virus cause many diseases to plants, animals, and human beings. Virus means poison. There is no any beneficial activities, only the damage, the disease causing organisms, making the disease. It causes disease plants, animals, and human beings. Let's study about that characteristics of virus. Already we know very well the character, two character, living character and non-living character. The living character, they respond to heat, chemicals, and radiations. They reproduce inside the host cells. Outside of the host, it cannot reproduce, it cannot multiply. It is inactive. They reproduce inside the host cells and produce copies of themselves. So these are the two living characters, respond to heat, chemicals, and radiation. They reproduce inside the host cells and produce copies of themselves. The non-living characters, they are inactive when present freely in the environment. The environment, when it is outside the host, it cannot make any function. They can be crystallized and stored for a very long time like other non-living things. This is the two non-living characters and living characters. This is a bacteriophage complex structure. The polyhedral head is here, then collar, tail, the complex structure of bacteriophage, T4 bacteriophage. The influenza, spherical in shape, and tobacco mosaic virus, helical in shape. Next, we'll study about bacteria. Bacteria are single cell prokaryotes. So prokaryote cells without nuclei, but nucleoid is present. So what is the meaning by nucleoid? The nuclear membranes are absent. That is what? Nucleoid. Nuclear membrane absent. That is nucleoid. Nucleoid is present. Nucleoid without nuclear membrane. So they are considered to be the first living organisms on Earth. Bacteria are grouped under the kingdom Monera. The study of bacteria is called bacteriology. And previously we studied about that study of virus, so virology, study of microbes, microbiology, study of virus, virology. The study of bacteria, bacteriology, study of bacteria, bacteriology. The size of bacteria ranges from 1 micrometer to 5 micrometer. Bacteria are of two types of bacteria based on respiration, aerobic bacteria and anaerobic bacteria. Aerobic bacteria is require oxygen for its respiration. Anaerobic bacteria does not require oxygen for its respiration. So based on bacteria respiration, it classified two types, aerobic bacteria and anaerobic bacteria. Then cell structure of bacteria. A bacterium has one outer covering known as the cell wall. The bacteria outer covering is cell wall. The nuclear materials represented by a nuclei without nuclear membrane. Without nuclear membrane, the nuclear material, a nuclei, that is circular DNA. The extracircular DNA is present. This extracircular chromosomal DNA called plasmid. Plasmid is present in the cytoplasm. Protein synthesis is carried out by 70S ribosome. The ribosome is called as protein factory of the cell. So it synthesis protein 70S Swartzberg unit. This means the Swartzberg unit. 70S ribosome is present in bacteria. This is prokaryotic bacteria. Other organelles, mitochondria, Golgi complex, endoplasmic reticulums are absent. So flagella is present. This flagella is help for locomotion. Locomotion means to moving from place to place. Let's study about the classifications of bacteria based on shape. Bacilli, Spirilla, Cocci, Vibrio. Bacilli, rod-shaped bacteria is called bacilli, Bacillus anthracis. Spirilla, spiral-shaped bacteria. Spiral-shaped bacteria, Spirilla, Helicobacter pylori. Then Cocci, spherical-shaped or ball-shaped bacteria. They can stick together in pairs. If it is joined to Cocci, Diplococci, then form a chain-like, continuous chain-like structure, Streptococcus, or occur in bunches like a grapes, that is Staphylococcus. Okay, so Cocci, single Cocci, two Cocci means Diplococci, then continuous chain, Streptococcus, then clusters, groups, bunches, that is Staphylococcus. Then Vibrio as a comma-shaped bacteria. Comma-shaped bacteria, Vibrio cholerae, Vibrio. These are the four types based on the shape: bacilli, rod shape; Spirilla, spiral shape; Cocci, spherical or ball shape; and Vibrio, comma-shaped bacteria. Then the bacteria classified based on the arrangement of flagella. So flagella is a locomotive organ for bacterium. So based on that number of flagella, arrangement of flagella, it classified monotrichous. Mono means one, single flagella at one end. So monotrichous bacteria, Vibrio cholerae, Vibrio cholerae. Lophotrichous, tuft of flagella at one end. Example, Pseudomonas, Amphitrachus, top flagella at both ends. So amphitrachus, lophotrachus, top flagella at one end. It's a lophotrachus, amphitrachus, top flagella at both ends. So we can see here amphitrachus, the two end also the flagella present. Lophotrachus at only one end. Peritrachus, flagella all around. Flagella is present all around the surface of bacteria. The peritrachus. Atrachus without any flagella. There is no flagella. It's known as atrachus. Atrachus, monotrachus only one flagella. It's present only one end. Amphitrachus, two end also. Presence of flagella. Lophotrachus, bunches of flagella, top flagella present at one end. All around the surface flagella present. Peritrachus. So the bacteria it classified based on the structure, bacilli, spirilla, cocci, vibrio. Then based on the arrangements of flagella, monotrachus, lophotrachus, amphitrachus, peritrachus, atrachus. Next, bacteria, food, preparation of food. Bacteria get their food in many ways. Photosynthetic bacteria. Photosynthetic bacteria make their own food with the help of chlorophyll pigment, for example cyanobacteria. Bacteria that live in host environment use chemicals, ammonia, hydrogen sulfide to produce their food instead of utilizing energy from the sun. Photosynthetic bacteria can use the energy from the sun, but chemosynthetic bacteria cannot use the energy from the sun. So this kind of bacteria, it is called as chemosynthesis bacteria. Chemosynthesis bacteria. Some bacteria exhibit a symbiotic relationship. For example, Escherichia coli, E. coli bacteria live in intestine of human being. It absorb nutrients from the intestine. So bacteria reproduce by method of binary fission and multiple fission method. Okay, this is about bacteria. Next, fungi. Fungi are a group of eukaryotic organisms. In bacteria, we have studied about that. It's a prokaryotic organism. Because of that, nuclear membrane absent, other membrane-bound organelles are absent. But eukaryotic means the nuclear membrane is present. Nuclear membrane is present. Is what? Eukaryotic organism. But lack of chlorophyll. There is no chlorophyll. Without chlorophyll, the fungus are present. So without chlorophyll, fungus cannot prepare their own food. It's depend for food other living and non-living organisms. So they grow in dark environments. They may be either unicellular, for example, yeast. Unicellular means which has only one cell, or multicellular. Multicellular means which has more cell. For penicillium, example multicellular example penicillium, unicellular example yeast. They are found in all kinds of habitats. All kind of habitats. Habitat means what? The growing nature. Nature is what? Habitats. They are included under kingdom, fungi kingdom. The study about fungi is called mycology. In previously we studied about the study about microorganism, microbiology. The study about bacteria. Study about virus, virology. The study about bacteria, bacteriology. Okay, study about bacteria, bacteriology. Now study about fungus, the mycology. Study about fungus, mycology. Some fungi are macroscopic. Macroscopic means very biggest one. For example, mushroom, colon. What? Macroscopic. There are around 70,000 species of fungi is present in the living world. Okay, here you will study about yeast, which is a unicellular fungi. You will study about multicellular fungi elaborately in chapter 17. See, listen, this is a yeast cell. If you are going to observe the bakery shop, okay, they are preparing more cake, bun, on very soft cakes and buns are prepared. How they are prepared? With the help of the yeast cell. With the help of yeast, they are preparing. So the yeast is help for fermentation. Okay, fermentation. What is the fermentation? Already the bacteria, we have studied two types of bacteria, aerobic bacteria and anaerobic bacteria. Here yeast is taking respiration in anaerobically. Yeast taking respiration anaerobically. The fermentation process in our mother, when she prepared the flour of idli and dosa, she prepared in the yesterday itself. Okay, tomorrow morning it become what? Very biggest, very foaming and is showing the soft nature of idli and dosa. How it's showing? Because of the fermentation. அப்படியே பொங்கி நொதித்தல் அப்படின்னு சொல்லுவோம். நொதித்தல். See, for example, the milk is become a curd. It's also one of your fermentation process. Okay, so yeast are found freely in the atmosphere. Yeast grows in all kinds of media containing sugar. The cell is ovoid in shape. This is what? Ovoid in shape is containing cell wall and a nucleus. The cytoplasm is granular and has vacuoles. Vacuoles are present. Organelles and glycogen and oil globules are present inside the cytoplasm. Yeast aid in fermentation. With help for fermentation, the help of the enzyme, thymase enzyme. So it respire anaerobically and reproduce by budding method, taking the respiration anaerobically and taking reproduction by budding method. What is meant by budding method, sir? It produces bud-like structure, small buds on the surface of the yeast cell. This buds, when it's get matured, it become a new individual yeast. Okay, this is information about yeast cell. Next, algae. So algae are very simple plant-like eukaryotic organisms. You can see the pond and lakes, the green in nature. They are found in moist habitats, only the moist place. Moist means ஈரப்பதம். Moist place only we can see. They are rich in chlorophyll and can be seen as thin film on the surface of lakes and ponds. Okay, lakes and ponds, very thin film is floating on the water. Therefore, they are known as grasses of water. Grasses of water is grow on the water. Okay, like a grass. They are autotrophic. Why it's autotrophic? Because it has chlorophyll pigment. It can prepare its own food by the way of photosynthesis, and they produce their own food with the help of the chloroplast. So chloroplast containing chlorophyll, green pigment for photosynthesis. The study of algae is called algology or phycology. Study about microorganism, mycology. Study about virus, virology. Study about bacteria, bacteriology. Study about all fungi. Study about fungi, mycology. Study about algae, algology or phycology. They are given some algae, Chlamydomonas, Volvax, Ulothrix, Fricilla, Alva, Hydrodicatylon. Hydrodicatyon. So these are the examples of algae, different types of algae. See, their size varies from 1 micron to 50 meter. Algae may be unicellular and macroscopic. Unicellular algae also present, and also the microscopic. For example, Chlamydomonas is a unicellular algae, or multicellular and macroscopic. Sargassum is very biggest algae, the macroscopic algae. Unicellular algae exhibit vary from shape, spherical shape, rod shape, and spindle shape, where the multicellular algae are in the form of filamentous chain-like structure or in the branches. Okay, in this section, the study about unicellular algae, Chlamydomonas alone. Only they are given in our chapter, Chlamydomonas. This is a Chlamydomonas. We have a question. Chlamydomonas is a plant or animal because it has a two character, plant character and animal character. Because it has a flagella, it can move from one place to another place like animal, and also it has a chlorophyll. It can prepare the food. The chloroplast is present. It can prepare food itself. So it has a character for a plant character. So it's a borderline of plant and animal. Okay. So Chlamydomonas is a simple unicellular motile freshwater algae. They are oval shape, oval spherical or pyriform the shape. The pyriform pure shape is a common one found in ponds, pitchers, and water tanks. They have a narrow anterior end and a broad posterior end. The cell is surrounded by a thin and foam cell wall made up of cellulose. This is a plant character. The cytoplasm is seen in between the cell membrane and the chloroplast. The cell contain large dark nucleus lying inside the cavity of the cup-shaped chloroplast. The anterior part of the cell bears two flagella. which helps in locomotion. The flagella, it's help for locomotion from one place to another place. The anterior part of the cell bears two flagella, which helps in locomotion. Two contracting vacuoles are seen at the base of each flagellum. The anterior side of the chloroplast contain a tiny red-colored eye spot is present. The Chlamydomonas exhibit a sexual and asexual mode of reproduction. Here it present that vacuoles, and also the eye spot is present. Eye spot is present. So it has a mixture of animal character and plant character. So we can consider this a borderline of plants and animals. Okay. Next, protozoan. The protozoan is a Greek word. Proto means first. Okay. Zoa means animal. First animals, true animals. The single cell eukaryotes, they are included under the kingdom of Protista. The study of protozoan is called protozoology. Protozoology is what? Study about protozoan. Study about algae, algology or mycology. Study about fungi, mycology. Study about bacteria, bacteriology. Study about virus, virology. Study about microbes, totally microbiology. Study about protista, protozoology. Okay, the protozoan is a very small animal. Okay, protozoan means what? Very first small animals. Okay, so they are found in ponds, ocean, in moist soil, and in the cells and tissues of plants and animals causing disease. They range from 2 to 200 microns, size 2 to 200 microns. Protozoan has specialized organelles. What are the specialized organelles? These organelles are used for movement, feeding, and other functions. Okay, the types of protozoans they are given here. The ciliates, presence of cilia for locomotion, for example, paramecium. Then flagellates, presence of flagella for locomotion. Euglena, this is a euglena. Then sporozoans, parasites, plasmodium. So these are the protozoans, small animals, protozoans. So the cell structure of protozoa, they are given here. Amoeba. Amoeba is a unicellular microscopic organisms. See, the amoeba, it does not get any definite shape. So we can draw at any shape. That is what amoeba is okay. Amoeba is correct. It is found in pond. Amoeba is irregular in shape. It has cell membrane, cytoplasm, and nucleus. It is a protozoan that moves by means of pseudopodia. There is no true leg. There's a false movement organs are there. False leg. There's a false leg. That is what? Pseudopodia. Okay, false leg. Pseudopodia means false leg. So here it is a protozoan that moves by means of pseudopodia. That's false feet. Latin word false feet. Pseudopodia are the extended part of cell membrane. The same cell membrane, it gets external part. Then for the purpose of movement, it helps to catch its prey. Not only for the purpose of movement, it helps the catch the food. It helps to catch the food. It surround the food particles and swallowing the food particles into the cytoplasm areas. It helps to catch its prey. Okay, maybe it can eat algae and other summer food. The body flows around the food particle and engulf means swallowing it forming food vacuoles, and contractive vacuoles are seen in the cytoplasm that help in excretion. The contractive vacuole after digestion, it excretes the waste materials because it's only one cell, unicellular organism. With the help of the vacuoles, the food vacuoles, then excrete vacuole, the contractive vacuoles are functioning into the cytoplasm for the purpose of taking food and excretion. Amoeba reproduce by means of fission method and sporulation method. Okay, during the parallel condition, you take a fission method, binary fission and multiple fission. The unparalleled condition is close to sporulation. Then it amoeba, and then next topics we'll study the next class. Prions, virions, uses of microorganisms. We'll study next class. Thank you, students. Thank you for watching. Thank you all.